The Difco Manual of Dehydrated Culture Media and Reagents for Microbiological and Clinical Laboratory Procedures. Difco Laboratories Incorporated, Detroit, Michigan. Introduction. Bacteriology emerged as a definite branch of science as a result of the monumental and immortal researches of Pasteur and Koch. When in 1876 Robert Koch, for the first time in history, propagated a pathogenic bacterium in pure culture outside the body, he not only established Bacillus anthracis as the cause of a specific disease in cattle, but he inaugurated a method of investigation of disease in general which is still pursued with unabated vigor. The decade immediately following Koch's epoch-making introduction of solid culture media for the isolation and growth of bacteria ranks as one of the brightest in the history of medicine because of the number of variety and brilliance of the discoveries made in that period. These discoveries, which, as Koch himself expressed it, came as easily as ripe apples fall from a tree, were all dependent upon and resulted from the evolution of correct methods for the in vitro growth of bacteria. The fundamental principles elaborated at that time, of which the most important was the introduction of poured plate method for isolation of pure cultures, still constitute the foundation of bacteriological research. Nevertheless, it has become more and more apparent that the successful attack upon the problem still unresolved is closely related to, if not dependent upon, a thorough understanding of the minutiae influencing bacterial nutrition. With a suitable culture medium, properly used, advances in bacteriology are more readily made than when either the medium or method of use is inadequate. The bacteriologist of today is therefore largely concerned with the evolution of methods for the development and maintenance of growth of bacteria, upon which an understanding of the biological and biochemical phenomena resulting from bacterial activity must largely be dependent. The chemical analysis of bacteria indicate that they are essentially water plants, the protoplasm of which contains chemical elements found in other types of plant protein. In order to build up the cells of bacteria in their anabolic phase, the protoplasmic elements must be found in the immediate environment. So vast is the problem of bacterial metabolism, and so numerous and diverse are the minutiae it includes, that a comprehensive discussion of its ramification is precluded here. It is the purpose of this discussion to present briefly only some of the outstanding developments in this phase of bacteriology, especially as they concern the evolution of new methods for the cultivation and study of bacteria. Almost without exception, wherever bacteria occur in nature, and this is particularly true of the pathogenic forms, nitrogenous materials and carbohydrates are present. These are utilized in the maintenance of growth and for the furtherance of bacterial activities. So complex is the structure of many of these substances, however, that before they can be utilized by bacteria, they must be broken down into simpler compounds. Such alterations are affected by the process of hydrolysis, oxidation, reduction, deamination, etc., and are the result of bacterial activities of primary and essential importance. These changes are ascribed to the activities of bacterial enzymes, which obviously are both numerous and varied. The processes involved, as well as their end products, are exceedingly complex. Those of fermentation, for example, result in the production of such end products as acids, alcohols, ketones, and gases including hydrogen, carbon dioxide, methane, etc. Abstract as such studies of bacterial metabolism may seem, their practical application is soon apparent. From these studies has come a better understanding of the nutritional needs of bacteria, and from this in turn has come the development of culture media, productive of rapid and luxuriant growth, essential requisites for the isolation and study of specific organisms.